आज मैं एक स्टोइक कोट पढ़ा। मुझे स्टोइसिज्म हमेशा वेदांता से मिलता जुलता लगता है। लाइक यू नो देयर इज़ अ फेयर चांस दैट डिफरेंट पीपल ऑप्ट different way to get the same truth. So it says that just assume that you are dead. You're already dead, and whatever you are living is the bonus. See, all these things, assumptions and everything. So, uh, everything is so simple at the same time so complicated that we never get to know what is the ultimate truth. Somewhere I read a quote, it says that death is just the beginning of life. Like this suffering ends, and then you really go in your original form. But these are just quotations. We do not have a proof for this. When we say proof or evidence, that is a fair way to judge something or to understand something. And when there is no evidence, then you have to go for a logical argument, right? So I decided that whenever I will work, I will do some logical argument about random things. Anything. The reason is very simple, that sometimes I feel that I get lot of ideas, lot of thoughts, different things, and while working in this dark alone room, I never get a chance to actually talk about it. First of all, who would listen about all this shit? People are busy either fighting Congress versus BJP, or secondly, a large fraction of society really, not able to do this kind of debates. Reason is very simple. People are looking for food. But how bizarre it is that we are, still struggling to get food, which is, if you ask an animal, they get food simply anywhere. So, thinking that in mind, I thought that my work is to logically understand the world. How does it function? And who understand the world, how does it function? One who get an ability to understand a small thing precisely and accurately, that person is likely to understand the world, the whole world. The reason is very simple, that you get an ability to think logically about any issue. So therefore I decided to talk about only protein, random things about protein. First of all, I am not an expert of protein. The purpose of making this video is very simple. I am in this alone single room, it's night time, I have nobody to talk. I have people to talk about it, but presently no, because I am busy in experiment. But my experiment can go on even if you talk through this video. So most of the time I'll not make a claim that what it is actually, but we'll make some simple argument about protein biochemistry, protein science and all. Without any reading, without any, proper prior understanding of what I'm going to talk about. It's rather simple that I'll just talk about random things, what I do, why I do, and this shit. So this is a tube. In this tube we have a solution of protein in a buffer. So now there are two questions in this statement that a simple mind would ask, that what is protein and what is a buffer. So the story starts from the very beginning, where people wanted to understand everything that is in front of him, and they found that most of the work happens in cell by protein. So you can call protein is the worker of our system. When I say system, I essentially mean this body and the cell system. So every cells have millions of protein molecules performing their duty, performing their work. So now again, third question is, what is work and how do protein do this? So everything, I have filled the protein in this needle that I am going to inject inside the cell, and as a result, that why do you have to inject something like a protein in the cell when you are saying that protein already exists in the cell? But those proteins are like millions of protein are present in the cell, and I cannot detect them very by specifically. I cannot detect a particular protein specifically. How would I distinguish where it is happening? So I label the protein with a fluorophore. Fluorophore is a compound which gives you the visual understanding that how things you can label it, and you can specifically there is a village in which there are a lot of people, and you want to study some few people of your interest. What would you do? And you don't know them by face because you do not have an ability to recognize their face somehow. Maybe you are so like you are observing them from a space station, and that space station has there is some resolution limit that you cannot identify their face, so you will tag them, like you will put some red gamcha on their or red muffler, so that you can identify them. That's why I labeled the protein. However, coming back to the original question, how do protein molecules do the work? So work is so what is work? So work is whenever something moves from one place to another place by against an already existing force, by some other force. So two things are required, like there is some external force and there is a displacement, that is called work. Now, inside the cell, these protein molecules keep on doing various functions, like they can carry oxygen, and they can also make glucose. Like there is a protein molecule called insulin, And this protein molecule insulin has an ability to push glucose molecules present in the blood into the cell. How do they do this? That is a different question, but essentially pushing glucose molecule from blood to inside the cell is a work. Again, carrying hemoglobin from one place to another place is a work. Also, when you say protein interact with another protein and then it gives you some function, it will also work. So basically, protein molecules are worker in the cell, and these molecules are made up of some definitely atoms. So basically carbon, hydrogen, oxygen, nitrogen, sometimes sulfur. So these atoms make this protein. So we don't generally call protein as a molecule, we call protein as macromolecules or biomolecules. The reason is clearly very simple, that it is made up of smaller molecules. It is a polymer. Protein molecule is a polymer, and this protein molecule is made up of various amino acids. Amino acids are different molecules, and these amino acids get linked with each other, and they form some sort of interaction that we in common language we call them bonds. So different amino acids make covalent bond with each other, and it makes a long chain of polymer. Now this long chain of polymer has high entropy because it can flip-flop, it can dance, and then everybody was thinking that as per second law of thermodynamics, higher entropic state is more favorable. Everything is more stable when it has higher entropy, higher randomness. So what happened, that people thought that this chain should be like this, flipping flopping and doing thing. But the problem is a protein molecule cannot perform a work, any kind of work, when it is open, when it is doing random dance. And it inside the cell, it works only when it is folded, only when it has some sort of shape. And different protein molecules are different because they have different shape. Some has shape like this, some has shape like this, some have shape like this, and consider these fingers are stretches of amino acids. Some are various bigger protein molecules. And this is how protein molecules function. And different amino acid, different protein have different function because they have different shape, different structure. So it was there was an ambiguity that why do protein molecule first of all fold when nature, we call second law of thermodynamics, which is a law, and Albert Einstein said that thermodynamics is only science that will go for eternity, and there will be no violation of thermodynamic laws. Nature, everything has to work as per laws of thermodynamics. So scientists puzzled in the very beginning that why do protein molecules first of all get folded and reduce its entropy. That is the first question, that why would it takes a shape that has got lower entropy. you know, and when protein molecules form, you know, by joining different amino acids on ribosome, which is a protein machinery inside the cell, then it essentially forms a chain. It is making a chain like structure, you know, connecting different amino acids. And when it forms in a, when protein is synthesized, it has higher entropy. Then why would it, you know, change into a smaller you know, a folded form which has got the lower entropy? That was the first question. And second question was this, that why different protein have different shape? So by just analyzing different proteins, if you look at their sequence, so as I said, that protein molecules are essentially you know, macromolecules or a polymer of smaller amino acids joined by an interaction. We call them covalent bond. So when we say bond, is not a strict like structure. Bond is an interaction in which has you know, when two molecules are randomly doing certain stuff, and if they get associated and form bond, essentially they have lowered their free energy, which means that they are more stable. So that's it. People started taking it like a, you know, they started representing this structure by a line. So it's very important in the very initial phase of life to look at elements and atom as the way it is. Generally, when we say carbon, we think it's like this, and when we say oxygen, then we say like this, you know, as a circle, and we say fluorine, we assume it's like some antenna-like structure, but that is not true. The truth is, when you assume and think about an atom, any atom, carbon, oxygen, fluorine, hydrogen, anything, so you have to think it like considering the very basic science, that it is a spherical, it has a spherical shape. Essentially, if you go down, you find that they also behave like some waves, and electron is nothing but a probability function, and then you go in deeper, you find string theory and all. But on the basic level, we consider every atom as a sphere. So if you think about what is the size of a hydrogen atom, it's 0.5 nanometer, the radius and all. So it varies, and in nanometer range, the nanometer is 10 to the minus 9 meter. So that is the size of an atom. So why different proteins are different? The reason is very simple, that different proteins take different shape because essentially their sequence of amino acids are different. If you compare, the sequence of amino acids are simply molecules, and in nature there are 20 kind of amino acids. There are two other amino acids as well, but that is not very common. It's very, you know, rare amino acids. That's why we'll not talk about it. We'll talk about a general kind of amino acids, 20 types of amino acids that exist in nature, and they get connected with each other, making covalent interactions, and ultimately they form macromolecules. So just to clarify a bit, that if you take a moti of different color, 20 different color, if you take beads of 20 different color and make a mala or necklace, then same, there are 20 different color of beads that you can use to make necklace, and depending on the you know, sequence of color combination, 20 types of colors, you will get different kind of necklace, and these necklace gets folded taking different shape because of the chemical properties of these amino acids. Some amino acids are hydrophilic, some amino acids are hydrophobic, some amino acids are you know, charged, some amino acids are have got you know, some different shapes, some has hinge, some has some floppy things. These are details. So that is the first question, that why do protein molecules, so this is the first question, that why do protein molecules have different shape? different protein molecules have different shape because of the different sequence of amino acids in it. Number one. Number two, the first question, that why do these molecules, these macromolecules, protein molecules, fold at all when folding is not supported by law of thermodynamics. And let me tell you, this folding is very much spontaneous process. Spontaneous processes are processes which doesn't require any energy from outside source. Hence, it remained a puzzle for many years that why do protein molecules fold at all spontaneously without taking any energy from outside source. Like we see that there are many reactions happening going from disorder to order, but it requires external energy, like photosynthesis. In photosynthesis, you take 6 molecules of CO2, 12 molecules of H2O, and you link them. And when you link them, you form 1 molecule of glucose. So essentially, you are associating 18 molecules to form 6 molecules of glucose. So you are decreasing the randomness of the molecules. Means you are decreasing the entropy of the system, and it is possible. Why? Because it is possible only because it requires sunlight. So sunlight gives you energy to perform this work. But let me tell you, this protein folding problem does not require energy from outside source. So what does it require? It requires nothing. It is a spontaneous process. So then people looked at it on detail, and they found, so there are various arguments behind it, that why does it happen. But there is a well-known argument regarding this, and that we call hydrophobic collapse or hydrophobic interaction. So what is it? So people have found that you all are well aware. Let's say there is a hydrophobic oil that you put in the water. What happens? That water hydrophobic. So you put 2 oil molecule, okay, 2 oil molecule in the water, 2 oil drops in the water, and they will associate. Most likely if they are nearby. So hydrophobic things have an ability to go away from water and interact with each other. They don't like to get associated with water molecules. Now the question was that why are we talking about hydrophobic oil droplets while talking about protein? So as I said, that amino acids are of different color, different chemical properties. Some amino acids are hydrophobic, some amino acids are hydrophilic. And here's the concept. So if there is a stretch of amino acids, if there is a polymer of amino acids, then hydrophobic amino acids are likely to interact with each other. And this process is completely spontaneous. Means mixing of 2 hydrophobic, intermingling of 2 hydrophobic thing does not require energy from outside source. It happens spontaneously. Now why? The reason is very simple. If anything happens, it has to follow the second law of thermodynamics, that says that total entropy of system plus surrounding tend to increase for a spontaneous reaction, or you can say the same thing, that free energy is likely to decrease for a spontaneous change. So what happens, that hydrophobic, when there is a hydrophobic oil droplet in your water solution, in a glass of water if you put it, then hydrophobic oil molecules do not like to mix with polar water molecules. So what happens? So let's say this fish of mine is a oil droplet, and this fish of mine is another oil droplet. And since these molecules do not just, if you put a oil droplet, they do not just dissociate with each other and form smaller droplets and get spread in the water glass. Rather, what do they do is this, that they will be like this only, okay. And there are water molecules surrounding this fish of mine, which is essentially an oil droplet. So water molecules are forming a case-like structure around these hydrophobic molecules. And there is another droplet. You consider this fish of mine is another water droplets, and there are water molecules forming case around this oil droplet, right? Now what happens that when these two molecules, water droplets, when these two oil droplet come near each other, so what happens, this phase of droplet and this phase of hydrophobic oil droplet, they are hydrophobic things, and they would like to associate with each other. And why spontaneously, and why is this possible thermodynamically? Because when they associate, when they come near to each other, water molecules which was surrounding this oil droplet, that cage breaks, and these water molecules around the hydrophobic droplets dissociate from the oil droplet, and randomness, entropy of water molecules in around this which was present in the cage around the oil droplet, entropy of those water molecules increases. Same thing happens when protein folds. So their hydrophobic amino acids, they likely to associate with each other. So when amino acids chain interact with each other in a covalent bond, and when they interact with each other by covalent bond, and at the ribosome, they form at the protein synthesis happens at ribosome, then it forms like a stretch. So let's say next amino acid is hydrophobic amino acid tryptophan, another is phenylalanine, another is tyrosine. So this way. So what will happen, as soon as amino acids, hydrophobic amino acids will come, there will be water molecules, water cage around this hydrophobic amino acids. Okay. So if you look at the now if you again look at this macromolecule, protein macromolecule, then what would you see? Not with this eye, but with thermodynamic laws and with this some experimental evidence, we understand this now. There are always growing science, but considering strong theory hypothesis, which is hydrophobic collapse model of protein folding, we are argumenting on that theory only, which says that when these hydrophobic amino acids associate with each other, then this cage, water cage around hydrophobic residues, they just dissociate and they gain the entropy. So it is the gain of water entropy while protein folding reaction that compensate for loss of the conformational entropy. So there is a compensation. Now, since during the protein folding reaction, gain of water entropy is more than loss of conformational entropy, and total entropy hence increases. This is number one point that favors the spontaneous protein folding reaction. Now, second thing is this, that when bond forms, like when these protein molecules, the chain of protein, flopping chain of protein molecules, when they come and then when they associate, then interactions happen. All these non-covalent interactions, like hydrogen bonding, like van der Waals interactions, like ionic bonds. So these interactions happen, all these non-covalent interactions, essentially happening because of why do non-covalent interaction happens? It happens because electron, proton, these charged particles, if we call it particle, then these charged particles interact with each other such that it lowers the potential energy. Okay, so when a van der Waals interaction happens, essentially it lowers the free energy of that system. So when bond forms, as a rule of thumb, I can say when bond forms, there is a release of heat, there is a release of some energy, and when this flopping, a macromolecule, when they associate and form a macromolecular structure, then they release some amount of heat. This heat goes in the environment and it increases the entropy of air particles, and that also favors this protein folding reaction. However, so this is a simple argument. There are other various arguments regarding this, and there is still ongoing argument, because science never ends. It ends at an open ends, which is not an end. So, but these theories are not in limelight nowadays, because it has been more than 55 years now since the Fenton's experiment on protein folding. So now science have moved, and hence I will not talk about protein folding all the time, because we know a lot of things about it, and as I said in the early, my purpose is to understand how does universe work, how does things work, and in order to understand how things work, I think that you have to understand how a small system works. If you gain an ability to understand how a small things work, you will definitely get an ability to know how things work at all. You will get an ability to think logically that how things work. Anything you can, after you solve a small problem, you can give a lecture about Congress BJP, you can give a lecture about whether triple talaq is good or bad, you can give a lecture about NRC, you can talk about Trump and Biden, and a lot more things. So that comes as a byproduct, as a result of logical thinking. So in this video series, which is not planned, randomly I came here, I was getting bored, and this light of this room is supposed to be blocked, because light may photo bleach my protein sample. So most of the time I just switch it off, and when I entered the room, light was on. So I had to wait for some time, and I thought to just make some random stuff just to make myself busy, because when I'm not busy I think a lot about other things. So and I started making this video, and it seems that I can share some of my knowledge with you. So and hence just I thought that I should be making more videos on it. Why? Because I read that quote by Epictetus, that just assume that you're dead and everything happens after it, just it's a bonus. So considering that, I'm thinking that I'm already dead. I'm a dead man, devil, whatever. Jokes apart. So I want to make some argument about my small project in the next videos onward. And my project is to understand. So till now, people have been studying this protein thing. Till now means till 10 years ago. So people have been studying protein folding in a cell mimic solution, we call it buffer, which has pH similar to cytoplasm. Protein molecules have evolved to function and everything inside the cell. So and it was in the beginning it was difficult to study protein in the cell. Hence people used to isolate. People have all today I also do the same, that we make protein in bacterial system or any system, then we isolate, we purify it by a very simple process. Like if you have some stones in your grains, cereals, then what you do, you just keep it in some sheave, and then you separate things based on size difference. Or if you have some some light particles in a heavy particle, then you simply just you know you have different ways to separate things depending on various ways. So how people have been studying protein, that they have prepared protein in the bacterial system following the biotechnology genetic engineering, and after genetic engineering the protein, and they isolate, they purify, and then they characterize the protein, and then they study all this behavior of protein, thermodynamic behavior, optical behavior, and a lot of things. But now, since ongoing argument about protein folding, conformational dynamics, aggregation was getting to a conclusion, like there was a lot of evidence, lot of argument, and people some sort of getting an agreement with each other, and when you agree with each other, the fight stops, and when you don't agree with each other, then you have to come up with essential logical arguments to prove your point. It's not that you will hate somebody because you don't like their ideology or their belief system. It's very simple, that if you don't like somebody, you have to come up with a better argument to tell them that, see, this is the argument, because of that you are wrong. Okay, and a fair, honest person would never mind calling him wrong based on some evidence and facts. And that is happening in that is not happening in the present Indian political system, which should be happening, and that requires logical people in Indian political system. Nowadays it's spiritual, not spiritual, religious. Religious is I always think that religious is a bottom, religion is the bottom of spiritual understanding. Why bottom? Because all spiritual understanding have been summed up in a way that should work for layman people, common people. But the point here is that after getting agreement with each other, now we have come to a conclusion. We know a lot of things about protein behavior in the buffer. So, and I am telling this because I have a personal of course I want to put my science before other people's science. That's why I always advocate my work, and this is it, that people wanted to move from test tube buffer to the physiological environment, which is cell, and hence it's important to study protein inside the cell only, not in the buffer. So what to do, how to do? It's a big mystery. I have been doing some stuff. Ben Schuller have been doing some stuff. There are people who appreciate people who are going inside the cell. There are obvious caveats. There are obvious complications inside the cell. But how does it happen? Stay tuned. I'll come in next video. Thank you.